a big stage, huh? Good morning, Brisbane.
Good morning, Lynn.
Good morning. Yeah, I'm Lynn Langit. Thanks, Chris. For the rest of you who aren't awake yet, this is Genomic-scale data pipelines. And in addition to me, of course, is Dr. Bauer. And she's going to talk a little bit about some of the other people on her team as well, because we're representing the work of her and her team.
Hello, Brisbane. So I'm Denis. I work at CSIRO. So CSIRO is Australia's government research agency. And at CSIRO, we're really passionate about translating research into products that people can use in their everyday lives, with modern Wi-Fi, one of the examples. But the research that we do, it ranges from astronomy all the way to material science. My team is somewhere in the middle. My team focuses on human health. So specifically using cloud computing technology in order to conduct experiments that previously would not have been possible in human health using big data. So Lynn, how does big data look in your world?
Well, as Chris said in her introduction, I'm an independent big data cloud architect based in the United States and California, but I work worldwide. And I really enjoy working with the biggest data sets. I've done quite a lot of work with startups that work with YouTube remonetization, Twitter, Snapchat, that kind of stuff. And from a study that was published, these domains are often cited as the biggest big data projects. From 2025, there's a prediction that annually YouTube, for example, will have this volume of data, and Twitter and astronomy and so on and so forth. So basically eclipsing the scientific data except for one domain, and that domain is genomics. So you can see the same scale if you kind of are not awake yet. See that? See how it's eclipsing it? So it's truly the biggest big data problem, and for that, I'm extremely interested in working with bioinformaticians, and it's interesting to kind of think about where this data comes from. So to get us thinking about that, Denis is going to start to talk just a little bit about genomics before we get into the tech.
That's right. So all that data, it holds vast amount of information. So it's 3 billion letters, that's the human genome, and in there encoded is basically every aspect of life. So every cell has one genome, and it can give rise to all the cells in the human body, so being that organs, heart, liver, brain. So all of this is encoded in this one ginormous string. Talking about big data here.
And again, how wide is that string?
3 billion letters.
3 billion letters. So B, 3 billion letters. So what are the implications of changes in those letters? You have a couple examples just to get us thinking.
Yes, exactly. So it influences the way we look, disease risk, obviously, but also the way we behave. And as an example, for example, there's the hitchhiker's thumb. So when you all put up your thumb like that, sorry about that, the thumb like that.
Everybody,
the last digit, it can extend further in some people. So obviously, I don't have that hitchhiker's thumb, but you can look at you and the person next to you whether that thumb extends to the back. And actually, there will be 1 in 3 that actually have this thumb that extends further to the back. The other example that I have up here is the way we behave. And the example that I like to think is coriander. So there will be 1 in 6 in this room that are passionate coriander haters. Where are the coriander haters? So obviously, this is due to a certain mutation in a gene that makes you perceive the taste of coriander as being soapy rather than pleasant. And that's basically just one tiny change. So the genome really has a lot of control, which is, that is the fun side, but obviously there's a serious side to that as well, which is diseases. So the most famous genetic disease, widespread disease, is cystic fibrosis. So there is one letter change, one mutation in the genome that leads to this devastating disease of your lungs filling up with fluid and you can't breathe, and ultimately it leads to death. So the genome holds enormous power and enormous information, and that is the reason that more and more people are sequenced in order to really harness that power and understand what is the secret within each one of you.
Yeah, and in fact, because the price of sequencing is coming down, the slide that I showed earlier with the amount of data, that's reflective of what percentage of the population is projected to be sequenced by 2025?
Yeah, so 2025, 50% of the world's population. Now, when you think about that, obviously that means that most of the ones in the developed world will have been sequenced. And there are numerous initiatives. So there's the Queensland Genomics Health Alliance, where when you go to the hospital and you have a certain set of diseases, they actually will sequence your genome in order to find out what is the best course of the treatment for you in that space.
So because this is a technical conference, we're really now going to start looking at technical implications and technical applications of this. And at first, this is data. So the data that comes off the sequencer is VCF. It's a text type of a data, but of course it's much wider, the 3 billion letters. And then when you're studying populations, you have a large number of samples. So it really gives, this aspect of really big data. I mean, when you look at it, it's kind of non-intimidating there with the different fields. It looks like, well, why would this be so complicated? But it's the size of the data in terms of the processing. That's one of the first problems, yeah?
Indeed.
So in the application of this, you've been working with another research group to help the audience to understand how you're then processing this data. So how does that workflow go?
Mm-hmm. So what we just see in the text, saw in the text file, going to fix that in a minute. So what we just saw in the text file is basically the mutations in the genome. So where one letter in the genome looks different in you than it would look in other people. And in order to identify, whether that is a disease gene, whether it's just you normal, happy self, or whether it's actually a disease-causing variant, you need to compare lots and lots of data. So here we partnered with Project Mine, which is an international consortium to look at 22,000 individuals with a motor neuron disease called ALS, which is the disease that Stephen Hawking has. So here the task is to recruit these people to understand their genomic profile and to compare whether one individual, whether a set of the healthy individuals have a disease what is the actual genetic underlying mechanism. And this is basically the part in the middle that my team is focusing on comparing the DNA profiles.
So to be clear, the solution that we're going to be showing here coming up is actually already in production, and it's being used in ALS research, which to me is very, very exciting. So to understand what you're looking for, I'll just take this while they're fixing her microphone. You basically are looking at gene profiles, and what you're looking at is the letter that is common amongst all the affected, and so that would be the T, A, G, C, and basically you have two states. You have disease cases and you have controls. And you can see that it's usually not this straightforward, where it's just a single genomic variant and it's just so clean and able to see it because you've got thousands of samples. But this is a simplified version of what her team's goals are, to find these variants that seem to have significance because they occur over and over again. And I don't know if you want to add to that, but it just summarizing there.
Back in action, Lynn, I'm so proud of you. This is my slide, this is biology, and Lynn represented it beautifully. So obviously, human health, right, and genetic is something that anyone can understand, can represent, can really, because it's valuable, it's important to know that. So yes, basically what I wanted to say on this slide is that it's actually the same thing with the machine learning problem set. So the machine learning people out there, how many machine learning people are there actually? Okay, so in a machine learning environment, what you want to do is you want to spot the difference. You have your features, and which are the letters here that Lynn said is a difference between you and the person next to you. And you want to know what is the letter that is different in the cases compared to the control, so in the people with disease compared to the people without disease. And that's a classical machine learning problem.
So in order to address that, Dr. Bauer and her team built a library, it's written in Scala, called Variant Spark. And she's going to briefly describe it as we get into getting ready to show you a demo of it.
Yes, so Variant Spark really takes the raw genomic profiles, does machine learning on Spark, and we're going to talk about that in a minute. And then the output is something that researchers are interested in. So this could be, what is the disease gene that differentiates, that drives ALS? Or it could be something like a patient clustering. So in the healthcare system, you might want to find out, what is the group of patients that have a certain genetic property, and can we treat them differently? So can we give them a certain drug that affects them better than others? So an example here is warfarin. The warfarin metabolism can be different between people, and it will be a good idea to know, in which class do you fall when you actually take warfarin next time.
Yeah, and so I think it was interesting for me when I first came on this project of why Dr. Bauer and her team chose Apache Spark for the solution. And I've done some work in the Hadoop ecosystem in the past, but frankly, I hadn't done too much production work in Spark. So when we started working together, we talked about this, and I thought it might be interesting for you guys to just get her perspective. So why did you choose Spark first, and why did you choose not to use MLlib in Spark? You wrote directly a custom machine learning library on top of Spark core. Why did you make those technology choices?
Yeah, so we actually started off with Hadoop, because Hadoop back then was, the big thing. You use it and all your problems will be solved. The problem, though, is that our data set was so huge that with Hadoop not doing the in-memory caching, it actually took longer to run it with Hadoop compared to other methods. So we thought, that's not a good thing. Let's move over here to Spark, where it has the memory caching. It has the ability to actually interact with the the equivalent of a queuing system, if you're familiar with the high-performance compute environment. So we can actually manipulate and use the data in a bespoke library in a way that really deals with our machine learning algorithms that we wanted to implement. So again, we started with Spark ML because it was there. We wanted to use it. We just wanted to get the result out. I mean, we're researchers, right? We're not developers. Therefore, we just wanted to use it in order to find our disease gene. But again, the problem was that it did not scale. And this to us was actually quite gobsmacking in that, something that was developed out there by the big, IT community, big data gurus, and yet it would not scale to our genomic problem. And the reason for that is that we're dealing here with a high-dimensional data set. So usually when you do machine learning on your big data, you have lots and lots of samples, but you only have a small representation for each sample, so say up to 100 or 1,000 features. Whereas we're talking here 3 billion letters, and the difference between you and the person next to you is about 2 million differences. And when you have a large data set of 22,000, and that leverages up to 80 million variances. So we're doing a machine learning problem on a data set that has 22 samples times 80 million variants. So that is 1.7 trillion data points. And apparently, no one had a solution for that.
Well, you do. So speaking about that, you did performance testing when you were developing this library, and this is your initial results. So maybe you can talk a little bit about this.
Exactly. So when I said there was no solution out there, we tested that. Of course, we're researchers, right? And so what we've done is we looked at the accuracy. So how accurate can we actually predict? In that case, it was ethnicity. So we've taken samples and patient samples, and we wanted to predict the ethnicity of each individual. So the accuracy, how well we are, and how fast we could do that. And we compared these packages that are out there, one of which is Google's Planet implementation in Spark ML. Others are R, C++, and the last one obviously is H2O, which is one of the...
And technically what they did is they created splits. So you have this n-dimensional matrix, basically, and they split both this way and this way, which is very, very logical and kind of embarrassing that we, on the technology side, didn't figure that out. And she's already so smart on the biology, and now she solved our computational problem, so of course she gets the keynote stage. But yeah, I mean, this really is the only method to scale to 100% of the genome that I've seen. So it's a very interesting implementation. And potentially not only for bioinformatics, which is one of the reasons that we're presenting it here, because as we get more and more types of data, we have all these smart cities, IoT for for example, there might be other use cases for which we have 10 million features. So it's really interesting implementation. Now, when I met Dr. Bauer, they were working with an on-prem Hadoop cluster, and they were really wanting to scale this using cloud techniques to get cloud scale. And so I applied what I know about building pipelines, and this is just a generic sort of way of looking at building a cloud-based big data pipeline. So you have to define the business problem, look at the data quality, look at the candidate technologies, build and test minimum viable pipeline pieces, and then assemble the pipeline. And so we knew what the business problem was, and we knew what the data quality was. So where we started working together was looking at what kind of technologies we would use for ingest and clean, which is minimal in this because of the way the algorithm works, which Denis will explain, to analyze and predict, and to visualize. And so we've been trying out different implementations in different vendors. So to that end, one of the first areas that we needed to look at was, do we want to implement Spark, because of course Spark is core to this, via infrastructure. So do we want raw virtual machines that somebody has to administer and cluster and all that kind of stuff? And we pretty much discarded that because she doesn't have that kind of tech staff on her teams. So we're looking in the realm of platform as a service or software as a service. Now, interestingly, Spark in the cloud ecosystem, whether it's Amazon, Azure, or Google, is really more of a platform as a service. So you have Amazon Elastic MapReduce, you have Google has their partially managed Spark clusters, and Azure has same. Now, there is another player out there, and that's called Databricks. They're the commercial player around Apache Spark ecosystem, and they have something kind of between platform as a service and software as a service. So they have, you buy Databricks units, which technically is either EMR cluster, custom EMR cluster from Amazon, or now newly an Azure cluster. But it's not purely like a serverless solution. That doesn't seem to exist yet. So in our evaluative process, we were looking at the couple of cloud vendors, and we're at the platform as a service area. One of the aspects of this is when you're building machine learning pipelines, it's really important to include tools that allow the scientists to have intermediate visualizations so they can judge the quality of the data. And these aren't specific to bioinformatics, but these are just general type of statistical visualizations. And so when we were building out our cloud-based solutions, we needed to use tools that would allow us to integrate these visualizations. And those are Jupyter-style notebooks. So you can think of them, and we'll have a demo here coming up, as a new kind of a terminal or a new kind of a REPL if you've not worked with them before, because you can put in code and get a result, but you also then can see the visualization so you can look at the quality of the data and you can continue with your experiment. I'm really getting hooked up here. So, okay, so now we're going to get up to the demo. So she works with disease conditions like cancer. But because this is a public demo, we can't show medical data. So we're going to solve another really important disease condition. Who's a hipster? That's the disease. Now, I'm kind of new to Australia. I've been here a few times, and I'm learning about Australian culture. And from what I hear, this is not really the best city to find hipsters, that you find them in the city down south. So this is a hipster from Melbourne. Just saying. So we're going to switch over to the notebook. And I'm just going to show you here. This is Databricks. This is a Jupyter-style notebook. So it's a web page. And basically, you have Markdown inside of here, and you also have code execution areas. So this is set up for Scala, which is how Variant Spark is written. And by the way, Variant Spark is available for any of you to poke at, look at. It's on GitHub. It's written in Scala, and the way you use it is you pick up the jar file, and then you bring the jar file inside of here, and you have a cluster. And this cluster is just a cluster of Amazon instances that happens to be running in the US, but you can run it in Australia. And then you associate the library. So here's the Variant Spark library, and it's just you click to import it. Now, the reason we picked Databricks is because it's quick to demo, but we are also comparing on other vendors. It just kind of makes a nice demo for this particular purpose. So now, let me go back here, go back into the notebook. And I've already turned on the cluster. Databricks makes available a single node cluster for free for an hour, and we make this notebook available, so you actually can try this out if you're into it. So the first section is Denis's. This is the disease that we're looking for with variant Spark. So what is this hipster disease? What is this part?
Yeah, so our hipster disease, synthetic data, is a model that is actually a real, how real complex diseases work, in that there are multiple locations in the genome that jointly influence the phenotype, and influence the phenotype in a way that is quite complex. So it's additive, it's multiplicative, it has different weights, and this is basically what I'm visualizing here with this formula. So therefore, we took 4 locations in the genome, we weighted them, we added them and multiplied them, and in the end, there comes out a score. So for each individual, and we took the 1,000 Genomes project, which has 2,000 individuals. So for each individual, we calculated this score, and we applied a threshold. If you're above 10, then you're a hipster. If you're below 10, then nah, you're fine. So this is the data set that we basically gave Variant Spark in order to rediscover or test whether we can discover the 4 locations that went into that score.
So you can think of this as kind of a test-driven development for a machine learning algorithm. Because again, kind of trying to bridge between biology and technology here. This is the expected from these genome samples that are used to take genomic samples and then label based on the scoring, like Denis said. If they have a certain threshold in variation in these genes, then they're labeled as a hipster. And then we run the analysis to see which chromosomes are most associated and variant, and we expect to get this score, 2, 5, 4, 7. Basically, we're checking for that to check the correctness of the machine learning algorithm. So inside of here's the setup. So we've already done this. We've added the library from the jar file, and this is published on a Maven repository. So again, you can use it, and as can other researchers if you know some bioinformaticians. And then we just create a cluster. Now in the community edition, you can only create a single cluster, and it's just so I have one running. You just click here, you put it in, you put the version, and if you are going to do this, make sure you use the version of Spark 2.2, and it's in the notebook, and Scala 2.1, because we have dependencies to these particular versions. And then once you've got the cluster, then you attach it, because the notebook then is the client. So the Jupyter Notebook is how you interact with the underlying Amazon machines. So as you're working through this, the first step in the workflow is to load the source data. So this is the data that would have come off the sequencers. And this is a Python code block. One of the cool things about this is in the research communities, programming languages are just tools. So in this particular case, Dr. Bauer's team was just more comfortable with the Python URL lib libraries to load the data, and then you just run the cell. And this is executing against the cluster and loading the data from S3 buckets into the Databricks file system, which is a type of Hadoop file system setup so that it can be then processed. The next step, once you load the sequencing data, is to load the variants. So I don't know if you want to talk about that at all while I'm running it.
Yeah, all right. So the variance, again, is the genomic profile, so the 2 million differences between you and the person next to you. So what we do is we load it in this HDFS type or like file system, and up it comes.
Yep, and you can see here's the loaded samples. And this is in Scala because we have no notation here. To me, it's been interesting working with the research community how they just throw all these programming languages together, and we reflected that in this notebook. Because for my past as a more traditional developer, teams maybe will use a front-end language and a back-end language, and that's been a learning for me that I see Python and R and SQL and Scala in this case kind of all in the same notebook. So the next thing is we're loading labels, and here we're applying the labels based on that formula. And so if I just run this, you can see from the samples from the 1,000 genomes, you can see like the first one is not a hipster, the second one is a hipster. And it's just labeling the data because as part of the algorithm that Dr. Bauer's team wrote, they're going to look at the labeled data and verify against it. Then the next step is to configure the analysis, and in their API, they wrapped the underlying machine learning implementation, which is a decision trees algorithm, and they call it the importance analysis because that terminology is more friendly to the domain, to bioinformatics. And in this case, they're spinning up 1,000 decision trees, which we'll talk about that actually a little bit after this, but it's how the algorithm works. Then once we initialize it, then we run the analysis. And I'm actually not going to do that because it takes 3 minutes. And then we see the results. Now, interestingly, inside of here, an important aspect of these notebooks or these new interfaces is not only documenting the workflow, running the different languages, and as you'll see in a minute, visualizations, but it's also integration of logs. So if I open this up, this is a nice integration into the underlying steps, because of course you've got these huge, huge amounts of data coming through, and when you're trying to properly tune your cluster, it's important that you have access to where the performance bottlenecks might be. So this is called the directed acyclic graph, which is the way that the computation is running, and you can click in and you can get, basically it's a log reader, so you can go to level of detail and you can find out where the computational overhead is so you can set up the appropriate tuning, which is really useful and important in workloads of this scale. So the next step, I'm going to turn it over to you, the math part here, which is the variant importance score. So kind of how the algorithm figures out which variants are most closely associated with the phenotype.
Let's leave it at that.
That's it? Okay. That's the math if you want it. So when you're done with that, one of the great things about the notebook environment is that you can visualize. So the notebooks themselves have visualizers. So I could put this back as data. But very commonly, you want to see some sort of visualization because you've got so much data. And incorporated in this version of the notebook, you have these types of visualizations available. And of course, you can plot them, which are basic and can be helpful. But what I find, and this is a SQL syntax on top of Spark that's being used here, is when I'm working with bioinformaticians, more often than not, seems like you guys tend to use Python because of the libraries, or R. So inside of here, this is the same plot, which is showing the most important variants, but it's a little bit fancier, and I have to brag because I'm trying to learn. I did this. I rotated this. This took a little bit of time. This is this right here. And all joking aside, this is a better visualization here.
Of course it is. Lynn.
Hey, all right, you can take the heat on the next visualization. So the next one is R. So this is loading it into R, and again, this is just flipping the graph so that you can see. Now, in addition to these visualizations, of course, these notebooks are basically web pages. So if you're cool like her, because not only can she analyze your data, figure out the best algorithm, she also can draw. Look what she did.
Yes, so this is the result. So luckily, remember what we wanted to do here is find out whether variance bug is any good. So whether variance bug can find in our synthetic data where we knew the truth, can find the 4 locations that we put in. And obviously it did, otherwise we would not show it to you. So this is the chromosome, so all our 22 chromosomes. And then on this axis, we're plotting the importance. So for each dot, so each dot represents a genomic location. It tells us how associated it was, how much it contributes to the actual hipsterness. And as you can see, there are 4 locations that are coming up and standing out like sore thumbs, while the rest of the information is squashed down in the noise level. And those 4 locations were the 4 locations that we picked, which is how we describe a hipster, which is having a monobrow, having fabulous hair or facial hair, having checked shirts. So the theory is that if you have checks, then that elicits pleasure in your retina, and there's a retina cell. And the last one is, of course, the coffee consumption. So here, your metabolism for metabolizing caffeine is higher. Therefore, you just have to drink more, right?
So I'm looking around, and I think there's less hipsters here than the other city we were in. I'm just saying. I don't know. Anyway, so the other thing that this type of a workflow enables, common in research, is you can document for reproducibility. Also, this can be checked into GitHub, and it can be permissioned, which is super cool. You have comments. You can publish it. And in her case, there's another genomic library called HALE, and so they did a comparative analysis that you can read if you're kind of into it. But the last thing I think is just really interesting, where they overlaid the results of the two, and I'll try to make it smaller so you can see it, of the two different libraries. And again, since this is your work, I'll let you kind of describe what people are seeing here.
Mm-hmm, mm-hmm. Okay, so the MIT method is a logistic regression method. So it's a standard way of how this data would have been analyzed if you don't use machine learning. So what it does is for each location, it evaluates whether that location is more associated with the disease than, more associated in the cases with the disease than in the controls. So therefore, it's sort of an independent test that you do over the whole genome. And as a result of that, what it does is it finds a lot of false positives because there are a lot of random locations that look exactly like the actual contributors. And the reason for that is that when you think about it, it's 4 letters in the genome. There are 3 billion of those in the genome, and therefore by random chance you can have features that have the same property as your label has. And this is coming up here. Whereas if you're using a machine learning method, then what you do is you train a method on the whole data set. Therefore, your chances of finding spurious information is a little bit reduced. And this is basically what we're seeing here, that we don't have that many false positives. Also pointed out here are the 4 actual locations in the genome. And you can see that our method found them in the right order, whereas the MIT method had them slightly mixed up.
So again, kind of abstracting this to a higher level, one thing that I'm doing quite a lot of work on, because I'm just really interested in it, is understanding how to visualize the results of machine learning. And this is an example, I think, of a really effective way to do it, because as machine learning becomes more pervasive in our big data workflows, it's not a skill that the majority of the people working in technology have. And so using visualizers is extremely important so that the results can be understood and evaluated. Now, to that end, we're going to switch back to the slides, and Denis is going to talk a little bit about how they implemented the machine learning in this invariant Spark.
Okay, so the machine learning method is a random forest. So you might be familiar with decision trees, where a decision tree is, the aim there is to split the features. So remember, the features are the actual locations in the genome. So it will pick a location in the genome and split the individuals on that. So if you have an A, you go in this bucket. If you have a T, you go in that bucket. And then it goes down to the next level. Well, all the A's, now let's pick another location. All the ones that had an A here, and in the other location may have a T, go in this bucket, and so on. You get the gist. And the optimization is the fact that at the end, you try to have your nodes, your end node, your leaf nodes, as pure as possible. And pure in that case means that the ones with disease end up in the same node, and the ones without the disease are in totally different nodes. And this is basically how you build a decision tree. A random forest is an ensemble of these decision trees. So in the end, you can sort of think about it that it's evaluating the whole space and trying to sort people, like put them into their own boxes. And that's basically what a decision tree is doing. And random forest is sort of the abstraction and the combinatorics of that.
Well, also random forest has an averaging function that helps to get rid of outliers, if I understand correctly, and also speeds up the analysis of the data because you need less data cleaning. You can just feed the data in compared to some other algorithms. So it's really important in her use case because she wants to get fast feedback. And so in some of the other implementations, there's a whole bunch of cleaning steps that are computationally intensive. And in this case, you kind of just generally throw the data at it and get the result, which really fits the use case. So in fact, not only can you do that, you can scale it. So you want to talk about scaling because that's super important here.
Yes, indeed. So you might have a small cohort of people, or you can have whole population scale analysis. And we needed to have algorithms that can deal with both ends. So to that end, we tested that if you have an increasing numbers of features, so this is looking at your a a reduced representation of your genome or the whole genome, which is basically this axis. And what you can see is that the runtime increases linearly. Similarly, if you have more samples, so if you have a larger cohort, so there's a number of samples that increases, it also increases, nearly sublinear even. And this really showcases that this methodology can really deal with small data sets as well as those high-dimensional, huge sample data sets that we might have. And actually, in terms of price, you can deal with that too. So a small, traditional old methodology analysis style would cost you $200, whereas if you go to the new methodology of, say, 10,000 individuals, that will be in the order of slightly higher, $9,000. That's still affordable.
Well, she must have a better budget than a lot of my clients do, because when I saw that, I said $9,000 is, we need to work on that. And so that's, kind of some of the work that we're doing, trying to figure out, can we use spot instances? Can we do reserved instances? Can we, how can we get cloud economies? Because that may be okay, but I like these fast feedback cycles where you can run whatever tests you need to run very quickly and very cheaply. And $9,000 is too much money. So we're working on that.
I like the sound of that.
Yeah. So speaking of things we're working on, there's some other things you guys are working on in the algorithm.
Indeed. So currently it's just mutation data, so it's genomic profiles, which is great. It's a good start. But when we look at the electronic medical records, for example, there's much more data in there. And that data is not categorical. Right? It can be continuous, it can be actually mixed of those. So therefore, what we want to do is extend the random forest, and the theory of random forest is extendable to mixed variables, continuous variables, and that's what we want to do. On the community end, and this is where Lynn says the smarts of the room comes in, is really that we want to cater for variant spark being used not only in the medical space, but also outside of the medical space, because it is actually agnostic of the underlying data. And therefore, the first step that we're doing, and this is partnered with Denis, who volunteered to actually implement the Python API, which we hope has a broader reach for the application. But also, the other big thing that is really close to my heart is that we find an application case outside of the life sciences, outside of the genomic space. So if you can think of a use case, come and talk to us afterwards.
For sure. And you can thank me for the Python API, because I'm a crappy Scala developer. And when I pulled down the library and started to try to work on it, I said, oh, no. If we want big scalability on this, maybe you guys are all Scala developers, but it was the first time I looked at it, and I said, please, can I have some Python? So if that works for you, you can thank me. So we have an additional project to talk about. We're talking about Variant Spark, which is a server-based project, but we're going to now kind of switch gears and talk about a second project that is in this genomics workflow. And to move there, we're going to, again, just do a little bit of biology, just a quick biology. Oh, by the way, you can try this out. So the slides will be up there. You can try it out. So what is this picture?
So genome engineering, CRISPR. You might have heard that in the general press. CRISPR is the way, or is a new technology in order to edit the genome of living organisms. And this really has the potential of bringing in a golden age of medicine, because with that technology, we can do things that previously were just not possible. For example, fixing genetic diseases like the hypertrophic cardiomyopathy. So this disease is actually 1 in 500 Australians have that disease. It's a heart disease where your heart wall increases, and the worst case outcome is a sudden cardiac arrest followed by death. So obviously, it's not a very good disease to have. And it's a monogenic disease, so that is the good news, which means we could, in theory, edit it out. And how close we actually are to that was demonstrated by a Nature paper, which was published 3 months ago, that demonstrated that you can do that in human embryos, and you can do that in 7 out of 10 cases. But that means, the flip side to that means that in 3 out of 10, it did not work. And when you think of this being your unborn child, then 3 out of 10 is just too high of a failure rate. So this is where we were coming in, and we're hoping to build, or we're building, a computational framework that increases the odds to make it work the first time, every time.
Okay, she's scary sometimes. It's a Google for genomic search is what it is, so they can find the spot, okay? That's what they were building, so they can find the edit spot. So in order to do that, you would need something that's burstable because you're searching in different locations and all over the place. So that's a typical kind of a serverless architecture on the cloud. So when I, again, was starting to work with these guys, I was thinking, oh, maybe I can work with some bioinformaticians and build out serverless because I'm building it in other domains and then they can get economies of scale and you only pay for the compute once it's accessed rather than the VMs. Well, they already did it. So she did it, and this is what it looks like. And I guess if you want to talk about some of the implementation, and then we'll show you what the app looks like.
Yes. So search engine for the genome, that's what it is, where researchers type in the gene that they want to target, and this framework comes up with the best option of how to actually target it. And the way that it does that, obviously, is it needs to search the genome. So at this stage, there is a lambda function, so this symbol represents a lambda function that is just a regular expression, right? So this lambda function is just a regular expression. It goes through the genome, it finds all the options that are out there. Now, not every site is created equal. Some of them you can edit really easily, others are just too hard, and researchers are just wasting their time if they would pursue them. Therefore, what we need to do is we need to identify whether they're any good. So here we have two workflows. One is to look at whether the site itself is good, but also, and this is up here, the interesting bit, is whether that site is actually occurring somewhere else. Because when you think of 4 letters, 3 billion positions, a site that might be 15 base pairs long, or letters long, they can occur at random. Therefore, if you have your beautiful spot over here, and it has 50 other locations in the rest of the genome, the editing machinery gets confused and it will start editing here, which is good, but it might edit over here in the genes that you certainly don't want to edit. Therefore, we need to make sure that the locations that we find are unique. And this is basically this part here. The problem, though, is that lambda functions have certain limitations. They are not very large and they can't run for very long. But a search in the genome, in the whole genome, is very large and it takes a little bit of time. Therefore, it just did not fit with the limits of the lambda function. Therefore, what we needed to do, and in the end this is a blessing in disguise, was that we had to parallelize this. So we had to partition the genome and search on these individual partitions and then collect the information back together. And this is basically this somewhat magic up here where we populate the SNS topic with the locations of the genome that it's supposed to search, and then the SNS topic is triggering a lambda function just for that location, which then deposits its results over here in the result table.
So what's interesting about this is her team attended an AWS summit where this serverless pattern was shown, went back and applied it to this problem. And as it says up there, it is one of the first serverless applications in research. It was published as a reference architecture on Jeff Bauer's blog. It happens to be on Amazon, but this pattern, of course, could be applied to any of the cloud vendors because they all have serverless. And it's just fascinating to me to see how Dr. Bauer and her team have taken advantage of the new cloud technologies so that they can more quickly get the information that they need. Now what this looks like is this is literally a screenshot of it. And again, I represent kind of the non-bioinformatics person. So green is good, black is bad. Okay, green is where you cut, okay? But you can see there's the section of the genome. So when I encountered this work of Dr. Bauer, I looked at it as a cloud architect and I said, okay, are there any bottlenecks? How's this thing running? And so what has come recently in the world of serverless architecture, and again, we just happen to be using Amazon, it's similar in Azure and in Google, is log readers. Because when you're working with serverless applications, you don't have root access to the machines. You can't SSH in. And so when you've got these workflows, you need different kinds of tools to be able to understand where the bottlenecks are. And so Amazon had released this log reader X-ray tracing tool. And I shared this information with Dr. Bauer's team, and we had some good success with it. So what did you find when you used X-ray?
Yes. So Lynn was saying we were the first to do this, which of course was our aim, but we did not have any other choice. Right? Serverless was the only way of making available to the world for free a technology that is so compute intensive. Imagine if we had a serverless cluster, it would have cost immense amounts, a normal cluster, it would have cost a lot of money. So this was the only way of making it available within the budget that we had, which means that every second in that workflow counts. The quicker we make it, the cheaper it will be for us, the more sustainable it actually is for us to maintain it. So therefore, our aim was to find the bottleneck, the one Lambda function that really takes more time than any others. So what we've done is we tallied them, again, researchers, right? So we tallied them, and we found that there's this particular function that takes an insane amount of time. And this is actually stuck in that on-target sensitivity, so where we evaluate whether that site itself is actually a good thing. And therefore, we had two Lambda functions that would evaluate those sites, being these two blue bars. And this is sort of an academic tool that we just used, and it's implemented in Perl, and it took a little bit longer. I mean, it's a nice result, but it just took too long, so we had to do something about it. And what we did about it, sorry, that doesn't work.
Here.
What we did about it is that we thought, okay, the way that it's implemented, we can do as well. We can do actually with a machine learning method. You probably find that not very surprising. So we used a random forest again and implemented this, the single Lambda function. And as you can see, this single Lambda function is actually quicker than these two, and certainly these two combined.
Yeah,
And so in the end, we managed to improve the runtime by 80%, bring it down from 2 minutes, which is not bad, but it's still quite expensive, to 30 seconds. And this means that it's much more sustainable for the research community to continue to using it.
Yeah, and again, a key sort of higher level thing is the log reader allowed the visualization of these various logs, which are just CloudWatch logs, and really kind of hard to parse. And so the pattern is, as serverless architectures become more mature, to really take advantage of the vendor tools, because before this was not apparent to you, and it was immediately apparent the first time you ran X-ray. So it's a good sort of win around that, so using the new tools. Now, using new is good sometimes, but not always. So what Amazon has released for orchestration of Lambda, because they don't store any state, and you saw the DynamoDB instances there for storing state, is a set of services called step functions. And they have correlation tokens. So if you know, like a BizTalk, for example, it allows correlation between these slices of compute. Now, the way that Dr. Bauer's team had been solving this was with that SNS topic, which is free. So of course, your vendor will come out and say, use our new latest, greatest thing. And you want to have some independent evaluation, because after looking at this, there was a cost to this, and it didn't add any value. So as X-ray was positive, step functions just were not needed, and actually you're not going to change the architecture there. So you don't want to just latch onto every new shiny around these serverless systems. You really need to test it out. So you've got some future directions for GT-scan. What are those?
That's right. So just like VarianceBug really benefited from the idea of a notebook where you can write your code, you see intermediate visualization, and you can share it with the person next to you or a researcher or a colleague on the other side of the world. Therefore, we figured we need to do the same thing for a GT-scan. So GT-scan is a web service where you manually interact with the GUI, but we also implemented an API. And as soon as you have an API, you can chuck it into the notebooks, which makes it instantaneously reproducible. And therefore, this is our way of doing that, of interacting with the Jupyter Notebook, GT-scan to use APIs. So the result can be retrieved within the notebook. So we fire off an API call. It tracks the information back from the web service, and it has it right there, and it can be interpreted or analyzed with a tertiary analysis of visualizing it or doing some other stats on it.
So the higher level pattern there is with this serverless architecture. What her team has done is not only use API Gateway to expose this as a website, but they have, in a kind of, I think, really innovative way, thought of tying this into the Jupyter Notebook environment for reproducible and shareable research, because that's how her community tends to work. So I just thought that was very interesting as well, and was new to me. So kind of pulling these patterns together for those of us who don't work in bioinformatics, because this is actually, honestly, the first project I've ever done, and one of the reasons that I'm giving this keynote is hopefully to inspire some of you. I worked on a lot of what I would consider socially meaningless Snapchat remonetizers and that kind of stuff in LA. And about a little over a year ago, a friend of mine got cancer, and she was unable to get personalized treatment. And I just made the decision that I was going to try to do some work in this area. And frankly, to be standing on the stage giving the keynote with someone as accomplished as her is pretty daunting. But I offer myself up as someone who has spent time. I've watched Illumina sequencing videos and tried to learn the bioinformatics. And when they were fixing your mic on that slide where I had to take over for you, I was kind of going crazy. But I got it, right? So the point is, you can learn. You can be part of these complex domains. So these patterns that we've seen, which apply not only to bioinformatics, but as these big data workloads come into our world. The first one is search, what we just saw with GT-scan, and it was a pure serverless. So again, we happen to build out on Amazon, but it could be on Azure, it could be on Google Cloud or whatever. You have got the idea of a data lake or file buckets, and then you have Lambda with API Gateway. This is very simple, elegant, and clean architecture. And in an Amazon world, that's kind of what it looks like. You may use step functions, you may not. The complex analysis, heavy lifting, that was done, or that is being done with a Spark server cluster. Now, one area architecturally that's really interesting, and I kind of alluded to this when we were talking about technology decisions, is in public cloud, there doesn't seem to be Spark as a service yet. There is this move towards serverless data analysis. Amazon, for example, announced a serverless Aurora in beta last week at reInvent. So the economies of serverless and the efficiencies of serverless clearly have worked for Dr. Bauer and her team. GT-scan is entirely serverless. So this is an area that we are highly interested in understanding what the vendors are doing. We don't see any Spark serverless at this point. We see Databricks they have Spark as a service, but they don't really have competition. So it's a really interesting area for complex computation. We're still server-based right now. Partially managed, but we're server-based. And that's kind of what this looks like. So when we were interacting with that notebook, it was just a big cluster. Now, the one we worked with had only 2 nodes, 1 master, 1 slave. But typically, you use like four slaves, I think, and 1 master, so a 5-node cluster. And then you interact with the notebook. So from the highest level, the way that for bioinformatics and by association, big data workloads of these sizes are addressed in this particular situation is Spark server clusters and custom machine learning, or serverless pipeline. So these patterns, I think, are patterns that we're going to start seeing in other domains because of the volumes of data. And I guess the last thing that I'll invite you to do as we kind of close this out is, if you have an interest in working in this domain, honestly, if I can do it, you can do it. And there's more need for more technologists, because she's got to find out where to edit the gene. I want her to focus on that, and I'll work on getting the price of the cluster down, because she's got some important work to do. So these are the 2 libraries, and I'm Lynn Langit, and
Denis Bauer, thank you.
Thank you very much.